you see the slide?
Yeah.
So
So
yeah,
let me, I'll just give a very brief intro and then you can take off.
sure.
Okay, so yeah, Dave Rovniak is currently at Bucknell University, and he did his PhD work at MIT under the mentorship of Bob Griffin, where he was working, I believe, on some cool quadrupolar and O17 NMR work. After that, he did a quick postdoctoral stint with Bob, and then took the long trek across town to Harvard Medical School and did another postdoc with Gerhard, where I believe is where a lot of the non-uniform sampling work started up. And I've heard you talk several times about non-uniform sampling, but today I think we're going to hear a little bit about some metabolomics. So take it away.
Awesome. Thank you so much. And is the sound still okay? Just to double check one more time.
Yep, we can hear you and we can see your slides.
Awesome. You know, just one comment, anybody who's ever taken the Green Line knows that that's a very long trek.
Absolutely.
So just had to comment on that. This is such an inspiring day, and so honored to be here, and thanks to the organizers for bringing this together. This immersion in such advanced NMR is a wonderful development opportunity for everybody here. Thank you all so much. So very briefly, a little bit about the context of this work. So I'm at Bucknell University, so I really resonate with what Nick said. We are in a primarily undergraduate situation, but we're also at an institution that has high research expectations and which works to create infrastructure and support for research. And so I really value being in that kind of a setting. And we also do have a limited master's program, and some of the work you'll see today is due to both undergraduate and master's students. And I'll try to do a good job of calling them out as we go. I sort of want to give you a tour of some of the things that, before we focus on metabolomics, tour of some of the things we work on. A career-long arc for me has been a rewarding interaction with several of my colleagues to study the biophysics of bile salts and their self-assembly properties and their guest host properties, which are just incredibly complex. Bile acids were, I think, some of the very first outstanding information on bile acids was done by NMR in the 60s with some really beautiful experiments that began to unravel the complexities of bile acids. And we still wrestle with bile acids today. They're a great intellectual playground for us, but we've learned a lot about them. For me, it's a career-long arc. I hope I'm nowhere near retirement for a while, but I've already realized I can't retire until we answer several more questions about bile acids. They're a lot of fun for us, and we even get a good excuse to use NUS on them. Thanks for mentioning the NUS work. We love to work on non-uniform sampling. That's a wonderful community of folks who, the people who work on non-uniform sampling generally do so on a volunteer basis. And so it's a really neat community who works on this from different perspectives. We've really ended up looking at it from the perspective of sampling, and particularly through lenses like this curious maximum of 1.26 T2. And I'm going to pause and see if I can put a pointer up. Okay, hopefully you're able to see a pointer. This curious maximum of 1.26 T2 really engaged our imaginations for a long time. It's due originally to a fellow named Jerry Mattson in the 70s. It's been rediscovered by many of us, and what its ultimate importance is anybody's to guess. But as you look at the development of signal to noise across an FID, you realize that weighting non-uniform sampling gives you the opportunity to achieve true signal enhancements. They're modest. They're generally less than a factor of 2. In fact, they're generally much less than a factor of 2. But that doesn't mean we don't want them. And thanks to Gary Martin, he worked with us on a fun project to look at 1-1 adequate experiments, both in conventional and homodecoupled variants, where we were able to use non-uniform sampling to enhance those spectra and see nice improvements. And you know, a lot of people sometimes say they're always worried about what they'll miss by NUS. My thing is I always worry what you'll miss by not doing NUS, the appearance of new peaks. One of the things that was fun was the leakage of weak 1-2 peaks came through with the extra sensitivity. And we've also proposed an approach for sampling, a quantile-directed approach that we think has some potential, and we continue to investigate that. gosh, there's so much proteins today. I admit I threw this in at the last minute because there's so much proteins today. We work with constraints, and for us, we are interested in isotopic enrichment on a budget. And there's a fun little trick where you can add 10% C13 glucose at induction, and when you measure the incorporated glucose in the final expressed protein, you find that it lands at about 20%. So that's a nice doubling of your glucose. This does so with excellent preservation of CC spin pairs, so that you can see good, do all of your backbone assignment experiments. And we just tested it by assigning ubiquitin with it. So I'm sorry, it's ubiquitin. But in doing so, we did notice something that in hindsight, we should have been able to predict. But this particular approach leads to a knockout of CACB connectivities for the branched chain amino acids I, L, and V. And actually, this is something that sort of got my mind thinking a little bit about metabolomics, because you really have to think about pathways. So the CACB pair is not preserved in ILV, and we see that. So it has an interesting editing effect. Just thought I'd show something that we haven't written up yet, and I'm not sure if we will or not. We'll see. Kind of a very cost-effective amino acid knockout experiment inspired by work that I think that's Ko Takeuchi and Gerhard Wagner did, inspired by some methodology they developed. There's a neat way that you can knock out amino acids. And this was something that we tried on a very, very challenging, very unstable 30 kDa protein that we ended up not completing the study on, but it was useful to do some methodology work on. And we were able to actually perform some of the assignments with the benefit of these knockout experiments. So that's sort of a quick overview of some of the things we've delved in. But for me, I've made a bit of a career shift. We still do some of those things, but I made a bit of a career shift and gotten a little excited about metabolomics, and particularly NMR and metabolomics, and why. I'm going to sort of talk about these actually in reverse order. I think the first criticism is to say that the evolving workflow of NMR metabolomics is that we're seeing a movement towards service facilities where you submit your samples. But nonetheless, NMR metabolomics in particular has not yet moved completely out of the research lab for a variety of reasons, including expertise in operating the NMR certainly, but also people interested in developing pull sequences. There are folks applying NUS in metabolomics and so on. I think it's just an interesting challenge too. I think, well, why do we get into anything? Well, that's one reason. I think for me, I was one of the ones waiting on the wings. There are now quite a lot of demonstrated successes. I think most people are familiar with trends with branched chain amino acids and diabetes. And I think many people who have heard about inborn areas of metabobanking. They got a head start on biobanking for a variety of reasons. And because of that head start in biobanking, they now have an extraordinary biobank, unlike any other. It's the world's largest genomic screening program in the world. And we're incredibly fortunate that they're in our backyard and that we're able to interact with them and develop relationships with them. And so we realized there's sort of a special access to a new type of sample. These are not prospective clinical samples, as the title of this talk has foreshadowed. These are standard of care samples largely. And what can we do with them? And that was a very fascinating challenge for many of us. Before I go on, I want to mention a fellow named Brian Irving. He was a research scientist at Geisinger who invited us into this work. He's since moved on to Louisiana State, I believe, in an independent position. But I just want to acknowledge him briefly here for inviting us into this. So we kind of warmed up to metabolomics because this is not solid state, not protein NMR. This is small molecule NMR, and that's a completely new world. And we kind of warmed up to it with some fun small projects. If you ever want to get started in a new field, start off with an educational angle. And one of the things we did was a fun educational lab for where students go out and purchase OTC health supplements, and we do some analyses of the small molecule components. And it was remarkably easy to find things like unlabeled caffeine, unlabeled niacin, and then particularly you could find not only Viagra, but you could also find derivatives of Viagra. So for example, in the top spectrum here, we see some of the telltale signals that are well known and well published for Viagra. And then we have a fresh set as well, telling us that we have both Viagra and a derivative present in the same sample. That's was a very fun project for the students, but also sort of fun for us, got us a little more interested in small molecule NMR. Another sort of warm-up project was to do identification of edible oils by PCA. We just did this in the context of showing feasibility. We did not intend this to be a research project, but we found that sort of some people have latched onto this in the research literature. We appreciate that very much, but just sort of want to share that this was intended more to show feasibility of using this in instruction and how well it works. So a couple of fun things that we did to sort of warm up to this. But now to grapple with doing something a little bit unique. As I said at the beginning, we're in a primarily undergraduate environment. And I'm not aware of anybody setting up a human serum metabolomics program in a primarily undergraduate institution. I found that challenge interesting, and I thought that was also a reason why we would want to pursue this. And in particular, our serum is going to start off in general as a BSL-2 fluid. And we're fine with handling BSL-2 fluids at Bucknell, I should add. But the decision was made to convert this to a BSL-1 project for several reasons. It does increase the degree to which we are comfortable having students interact with the samples, for example. We also have a multi-use spectrometer. We didn't really like the idea of putting BSL-2 fluids in the spectrometer. So for a variety of reasons, we decided to convert this to a BSL-1 project. And we do that with aqueous extraction, so organic extraction of aqueous metabolites, I should say. And at the moment, we've landed on a very well-known set of conditions. These have been widely published. You should look at some great work by Nagana Gowda and Dan Raftery, who have investigated this thoroughly. We've selected 2-to-1 acetonitrile for a lot of our work. Even though a lot of people are moving into raw serum, we're actually not feeling like we've lost out. By not doing that. We're really impressed with some of the perks of extraction. So it produces samples of just exceptional NMR quality. We're used to shimming these to very small line widths, less than a hertz, often much less than a hertz. We're impressed with the reproducibility of the method. It lets us use traditional standards like DSS and TSP. So raw serum often makes the use of traditional standards more difficult. So people are going into areas of new standards to deal with that. So we can use very reliable standards. Avoids interactions of metabolites with macromolecules as well, something that, obviously messes with your shimming, but it can cause broadening and make it difficult to identify your metabolites and so on. So one of the things that we wanted to do as relative newcomers is we wanted to both train ourselves, but also examine some of the fundamentals. And we realized that there were some things about extraction that weren't being looked at. So we just wanted to convince ourselves of these things. So we noticed that extraction with acetonitrile recovers ground truth. We did that in mock sera. We also used that to hunt for limits of detection with several test metabolites. And we found that in general, our limit of detection was around 1 micromolar. And that's for a 600 megahertz instrument running a room temperature inverse probe. We also just want to pile on to what everybody knows about NMR and metabolomics, that NMR and metabolomics, one of the nice features is how reproducible it is. And just to pile onto that more, we wanted to see that that was true with extraction as well. So we did 5 independent aliquots of a pooled sample, ran them all independently, and I was actually astonished by the close agreement that we had there. One of the things that we were a bit nervous about was seeing field creep in metabolomics, more and more people going to higher fields, and why not? It's a great idea. And so we wanted to better understand our platform and our capability. And so thanks to Penn State, who allowed us a little time on their beautiful 850 and cryoprobe. And we did a couple of tests where one of the things that really struck me is certainly both sensitivity and resolution increased. I was particularly impressed with the increase in resolution. And in hindsight, began to realize why sensitivity was not as strongly changed. These are salty samples. These are sera. They're salty samples. I think a good deal of work to optimize concentrations to deal with salt and cryoprobes would be important. So we left away with this feeling that we still had a platform that could detect a good number of metabolites with good sensitivity. A fellow named Matt Mealy really dedicated himself to some fundamental work that, first of all, pursued some questions we were interested in, but also, again, sort of deepened our expertise. This was kind of a silent phase of our entry into metabolomics, where we weren't talking about it for a while we were working on these things. And just want to point out some stability tests at 20 degrees and 4 degrees. These are all 4 replicates each. And we're watching how the metabolites change over time. And this is an extracted sera, which nobody had ever looked at before. And, you know, we were just curious, how long can we hold on to these samples? We got varying advice from different people about the longevity of samples, and we realized the only way to find out was to test it. And in general, most of these hold up pretty well over time. But we were intrigued to notice that there were some that didn't hold up as well as we might have guessed. So take something like valine, does very well at 20 and 40 degrees over long time periods. If valine is your target, you can be really hard on your sample. And phenylalanine, for the most part, held up pretty well. Pretty surprised that acetate fell apart at 20 so quickly, but refrigeration protects it. Glutamine fell apart pretty quickly, but is relatively protected by refrigeration, but it's not as much as you'd like. And you're starting to realize that not even refrigeration makes you completely happy there. And so there were a few metabolites there that basically were giving us about 2 hours from the moment of resuspending the dried pellet in an NMR buffer. We were getting about 2 hours where we could comfortably say all of the metabolites weren't changing. We were curious about frozen samples. And, you know, certainly intuitively, you would think that there should be good stability. And in fact, there is excellent stability. But if you're looking at this closely, you notice that after a period of about 6 months, there's a decrease in nearly every metabolite. And in fact, if you look a little bit more closely, the decrease is similar at -30 and -80. So wait a minute, that suggests it probably doesn't have anything to do with temperature. And in fact, what's probably going on here is that we're experiencing decrease due to freeze-thaw steps. And so that immediately taught us something that we need to think about. Biobank samples may be accessed more than once. We definitely need to put the spotlight on the number of accessions of frozen biobank samples. One of the other things that we did was, in order to earn the right to access this outstanding biobank at Geisinger, we actually went outside to a commercial supplier of serum and designed some straightforward tests around type 2 diabetes. Just put 10 in each cohort. And this was very non-clinical, very non-research cohorts, very limited metadata. Actually, to some degree, I think I was unsure if this was going to work. But in fact, it worked remarkably well. In fact, even better than expected. The very well-known result of branched-chain amino acids increasing with type 2 diabetes as well as with obesity came out very clearly. And in fact, other well-known markers came out as well, like tyrosine and others, and, you know, lovely PCA. And this was eye-opening because, first of all, we were then able to come back and say we've completed, you know, a successful start-to-finish metabolomic study, confirmed a well-known result. But this definitely got us a little excited about looking at sera that came from more variable backgrounds, from non-clinical populations. So this was very influential on us to encourage us to go in this direction. And let's sort of take a look at the uniqueness of standard of care sera and sort of just how bad things can be. So we've already talked about a number of accessions of frozen sera. Also want to point out that if you're doing standard of care sera, you may not have information that you need there. This is a draw that's taken solely for the purpose of providing care. So for example, if you're studying obesity-related diseases, you might be interested in C-peptide, but that's not necessarily going to be common in standard of care, so it's not going to be there. So you have to ask if you can pursue your research question without some of the pieces of information that might be missing in standard of care sera. So first of all, phlebotomy is, of course, a big umbrella for a lot of things, the person doing it, their techniques, and so on. So there's certainly variation there. Prospective clinical studies sometimes use a single phlebotomist, for example. The time of draw is no longer regulated. This is, when people can make an appointment to see the doctor is when the time of that draw happens, which means that fasting is not going to be particularly well regulated. Certainly everybody's instructed to do a 12-hour fast, but it doesn't have the kind of monitoring and compliance that you'd see with again, with a prospective study. You can't guarantee that all of your serum separator tubes or any of your other hardware come from the same batch. That's a practice in some studies to use single batches of your hardware. You may have to tolerate hemolysis in your sera. And also, you may have to deal with folks taking OTC supplements. We'll see some of that whole variety. Diet monitoring is something that you lose. Prescription drug compliance is something you lose. You will have multiple raters for the histology. You're collecting samples over many, many years with different times of storage. So this is how bad it can be. And so this is why we went into this a little cautiously to see what would happen. So overall, this is sort of our workflow. We validated our methodology, we validated the science, and then we went after our hypothesis. And the disease that was sort of partly chosen and partly just fell into our laps was NAFLD, non-alcoholic fatty liver disease. Probably many people have heard of this. 25 to 30% of the US population has some stage of NAFLD. That's not just the US, that would be Canada, Europe, that's all Western countries. It's considered a condition of Western lifestyles. Well, we'll get into the stages in a moment, but briefly, biopsy is the gold standard. But the problem is biopsy is invasive, risky, expensive, but also there's no way we can give a liver biopsy to 30% of the US population for so many reasons, including ethical reasons, when the majority of them don't need it. Yet the big question, the clinical dilemma of NAFLD is, of the 30% of the US population that has steatosis, a fatty liver, what percentage of them go on to fibrosis? Well, it happens to be about 10% of them, so approximately 3% of the US population is advancing to fibrosis. So you have this challenge of figuring out how many of the 30% are advancing into NASH and fibrosis, the advanced stages of NAFLD, and that's a big clinical dilemma. Steatosis is mild, it's difficult to diagnose. And generally, there are a few different ways of showing the spectrum of NAFLD. So steatosis is the beginning, it's a fatty liver. People often make a comparison to foie gras, not to be flippant, but rather because it's actually very interesting. Geese don't advance beyond steatosis, so why do we? It's a remarkable question. Some people are tackling that from a research perspective even. But generally, a fatty liver is your first stage. Then a condition, very, very broad condition called NASH takes over. And NASH tends to be described in two stages. NASH can begin with inflammation, and then it can go into fibrosis. And fibrosis, I believe it's these darker strands are showing us fibers that are forming. And finally, it can advance to cirrhosis, and actually even worse, cirrhosis can advance to hepatocellular carcinoma. NAFLD is crushing the US medical system, and dealing with it is one of our greatest medical financial burdens. We actually don't know how to deal with the ever-increasing numbers of people seeking care for NAFLD. It includes things like indications for liver transplants. So this is an enormous socioeconomic problem. Okay, I'm sorry to show you one of these tables that you always see in metabolomic studies. These are three groups. I want to draw attention to a few things that I think are important. These are morbidly obese female subjects. So these are BMIs all well over 40. And in particular, BMI did not change in this study. Now that's interesting. This was a convenience sample, so that was not by design. That simply happened. Also, type 2 diabetes actually did not change in this study. And again, that was not by design, that just happened. And those are a couple of things that are going to be seen to be important in just a moment. It was fortunate to be able to designate all of these as sera that were not previously accessed, so to deal with that one problem. To be fair, I think we're willing to work with that. I think we accept that as one of the sources of variation, to be fair. But it's very nice that in this study, sera were not previously accessed. Notice that insulin resistance did change. That's a hallmark of non-alcoholic fatty liver disease. So we hear insulin resistance, we think type 2 diabetes, but it's also a hallmark of NAFLD. And in fact, so is HbA1c. There are well-known markers of NAFLD in addition to in the clinical regime. They include alanine aminotransferase and aspartate aminotransferase. They increased very smartly, confirming that these groups are indeed progressing as expected, but mainly these groups were determined by histologies. By the way, one thing that we did is, knowing that we have a 2-hour window for stability, we first of all concentrated samples for Shigemi tubes, and we did 1-hour acquisitions to get the best sensitivity that we could. So this is a curated, non-high throughput study trying to leverage the advantages of having an operator at the spectrometer. And when we started on this, we were so excited, and then something frankly very upsetting happened. We saw a protein background in our first samples, and here's this blow up in the aromatic area, for example, and then you can see it all along in the aliphatics. Frankly, this was upsetting. This is what we had spent 3 years in the silent phase working to avoid. This shouldn't have happened. The DSS line is horrifying. You may not be able to tell, but it's horrible. And I remember telling the student who got started on this, Matt Taddeo, really, really outstanding student, careful scientist. I said, this could end as quickly as we started it, because we're working with donated human samples. We can't mess around with this. This has to be done right. And I asked Matt, go through this really carefully, look for anything that's important. I'll never forget that about an hour later, he walked into my office and he said, I think I've seen something. And I'm not sure if I would have seen it. And so I always want to be careful to credit Matt for his care and his great eyes for seeing this. Normal serum is going to have this yellow appearance that you see here. But when we are talking about the extraordinary BMIs of the subjects, such as in this study, it's common to see lipemic serum, milky serum. And it doesn't have to be milky to be lipemic, but many lipemic sera appear milky. And what he showed me was this remarkable tube right here, where if you see that fuzzy line there, that is not a graduation on the Eppendorf. That's a phase separation. And so I get to continue the theme of the day of phase separations that Lewis started us off with. And we realized that even though acetonitrile and water are miscible in all proportions, we were actually getting a liquid-liquid phase separation in these samples. And as soon as he showed me that, I just remember all the blood returned to my head, and I said, okay, we can do this. So this is where maybe it helps to know your PChem. So again, Lewis scooped me. I thought surely nobody else would talk about an upper critical point today, but we postulated that this was going to have an upper critical point. We postulated that we were down in the 2-phase region. And by the way, one of the things that helped confirm all of this is we would selectively remove the top phase and just analyze that. And it was a mess, full of lipids and proteins. And so everybody remembers their Lever rule, right? From physical chemistry, good memory, right? So from the Lever rule, that's completely expected. So these 2 phases are very messy phases. So what we hoped was that we could take this out of the 2-phase regime above what we hoped was the upper critical point, and no higher than room temperature, because obviously we don't want to risk the integrity of any of the metabolites. And so luckily, all it takes is a 5 to 10 minute step at room temperature to create a single phase, which then appears to pellet the proteins quite well, and to give really nice baselines and really clean spectra. So That was a huge relief. It was one of the most upsetting points in my career to realize that we had ruined about 6 or 7 or 8 human serum samples, but I think we learned something interesting about it along the way, and we've shared it for the rest of the world to learn from. So jumping to the answer, that's sort of how metabolomics works. You run all your samples, you run your stats, and you jump to the answer. This is not PCA. This is PLSDA. This is supervised clustering. So this clustering knows about the groups already. And I show this particularly at the start to show that we're really seeing evidence for strong intra-group variation. This generally suggests that we're working with non-clinical sera here that have many of those sources of variation that we looked over. Nonetheless, there's some really encouraging things here, particularly the control group is actually separating somewhat well from the very severe fibrosis group, and the steatosis group is approximately in the middle. So this is not the end of the world, and what this means is we just have to drill down and look at the means and take a good look at the means. We got about 50 metabolites, and credit that particularly to these very long runs that allowed us to get good sensitivity and to curated shimming to make sure we got excellent resolution on all of these. And used one rater. By the way, this is what you'll see in sera like this. You'll see acetaminophen, caffeine, aspirin. You'll see it all. And let's zoom to some of the findings. In particular, we're able to demonstrate inhibited fatty acid oxidation in steatosis. So that's through the decrease in ketone bodies. Ketone bodies are going to be acetone, acetoacetate, 3-hydroxybutyrate, which some people call beta-hydroxybutyrate. And we see a very clear decrease on progressing to steatosis here. And I want to stress that because we're going to come back to that. So in this data set, the ketone bodies decrease on steatosis and they stay decreased. Acetone decreases in steatosis and stays decreased. And you might be saying, hey, wait a minute, how did that acetone get in there? That's actually a really interesting question. These are extractions that finish with a few hours on a speed vac, and acetone is surviving the speed vac. In fact, we spiked this just to convince ourselves that it was acetone, and it was. We're actually not the first to see this. It's been reported by others, and in this particular study, we think acetone may be coming along because of the high lipid content is very protective and may be holding on to some of the acetone and protecting it from the speed vac. So. Alanine increases quite smartly. In fact, it's one of our most interesting metabolites from this work, and we have some neat things to say about it. If alanine increases, it's nice to see that pyruvate does too. Alanine increases particularly on the transition to simple steatosis, and that's a little foreshadowing for something that's going to come up in a second. Because as soon as you hear alanine, you think of a really important enzyme for non-alcoholic fatty liver disease, alanine aminotransferase. And it's a good biomarker for NAFLD and its progression. One of the things that we were very surprised to notice is that alanine and ALT were uncorrelated. And we thought that was very unusual, but that's going to lead actually to a hypothesis. But let's also see how they're uncorrelated, because how do you have two things that are strong markers, yet they're uncorrelated? And the way that works is alanine is particularly strongly indicating steatosis and weakly indicating fibrosis. On the other hand, ALT is more weakly indicating steatosis in this data set and more strongly indicating fibrosis in this data set. And that's particularly the insight as to how this is happening. This actually is going to put the spotlight off of just the alanine cycle. This is actually going to put the spotlight onto the urea cycle and get us to think about that in the early stages of NAFLD. So in particular, here's a common graphic that you see associated with the alanine glucose cycle, sometimes called the Cahill cycle, which shows that alanine is a nitrogen shuttle in the body, circulates between the muscles and the liver, and that glucose can certainly put pressure on the generation of alanine. But the key thing is, although sometimes when you look for the buildup of metabolite, you look for pressure that builds up that metabolite, you also have to look for constriction. You have to look for something that represents a bottleneck. And the key place where a bottleneck could occur is if we have a dysregulation in the urea cycle. Now, we're not the first to suggest this, but this is actually an emerging pathology for NAFLD. It's relatively recent. It's been considered particularly in mouse models. We think the lack of correlation between alanine and ALT is supporting that it's disruption of the urea cycle that's actually causing an initial buildup of alanine in early stages of NAFLD. And then in later stages, in fibrosis really represents liver damage, we start to see an increase in alanine aminotransferase in response to that. So this doesn't prove this by any means, but it's consistent with the emerging pathology that NAFLD involves urea cycle dysregulation, but also we're able to propose that it happens early in the disease in steatosis. So when do we inhibit fatty acid oxidation? Now, that's turned out to be a tricky question because there was a fortuitous study that was remarkably close to what we did, certainly some differences too. But they portrayed in particular that ketone bodies decrease in fibrosis. And I want to break this down and first draw attention. I know I hate talking through a table. It's terrible. Figures are much better. I want to point out that in this study in Finland, branched chain amino acids and tyrosine, known markers of type 2 diabetes, increased weakly but significantly. They did not increase in ours. Type 2 diabetes increased actually strongly and significantly. However, type 2 diabetes did not increase with us. And in fact, I think they encountered some difficulty recruiting subjects into the steatosis group, and I think that restricted things as well. But one of the biggest clues is it's a reality of NAFLD research that most NAFLD research has to happen in a background of T2D progression. So that's just a reality of doing NAFLD research. But something particularly interesting jumped out to us, which is the ketone bodies actually initially spike in the steatosis group before eventually decreasing. In contrast, we go straight to the decrease. And in fact, it's remarkable how similar our numbers are in overall magnitude. Now, that actually happens here as well with acetoacetate, one of the other ketone bodies. We don't see that as, we didn't profile that as confidently, but nonetheless, it spikes strongly in their steatosis group before undergoing a strong decrease in NASH and fibrosis. And what we're realizing is this huge spike in simple steatosis of ketone bodies is consistent with having individuals with strong ketoacidosis. And when we consider that T2D is increasing across their three groups as well, we're proposing that the steatosis group was confounded with type 2 diabetes and with ketoacidosis. And so we feel pretty comfortable claiming that inhibition of fatty acid oxidation does start in steatosis for those reasons. I think one thing we just want to sort of say is a broader outcome. We're pleased to see that the metabolites that distinguish the steatosis group are different from those that distinguish the fibrosis group, really telling us that the progression from steatosis to fibrosis is a really big metabolic shift. This is not necessarily a monotonic progression. There's a major metabolic shift that takes place. Wanted to jump ahead to exploratory classification, which we think is a good idea in this particular study because we do think we're working with populations that are broad and which have a lot of intragroup variation and which might be representative of broader populations. So we wanted to do some exploratory classification. We don't have a larger validation set. I should say, to echo comments previously, we have that underway, and if it wasn't for COVID, I'd probably be telling you about the validation set right now. But unfortunately, I can't tell you about that right now. Still underway. But we're pleased to see successes on the order of 80s, and in some cases even high 80s. And particularly what we want to draw attention to is by taking a small number of metabolites and combining them with even just one clinical value. Adding one clinical value can always give us a nice increase in classification success. And we're particularly interested in the combination of NMR-derived metabolite levels combined with strategic clinical values. And so we think that's important. So where this stands is we don't see branched-chain amino acids increasing in this work. And in fact, a recent paper just this year came from Dan Raftery's lab, did a related study, slightly different stages, but they did not see BCAAs change either. And so we found that really encouraging. And we think that when people are seeing BCAA changes, it's confounded by T2D. In fact, to investigate this a little further, we had enough, just barely enough in the fibrosis group. We were able to divide them up into two subgroups of early and late fibrosis. And remarkably, branched-chain amino acids actually all decreased with very strong significance. Now, that's really interesting because that's not actually a complete surprise. This is something that other researchers have seen particularly strongly in cirrhosis and somewhat in fibrosis. And so we're encouraged that we're able to actually strengthen that finding in fibrosis, that decreasing BCAAs do agree. Furthermore, decreasing BCAAs in fibrosis are correlated with risk for advancing in NAFLD. So we're really encouraged by that. We think that's a good validation of the data set, but also enhances the importance of decreasing BCAAs during fibrosis. So I want to sort of get on to the potential and the pitfalls because I promised that we'd get there. Feeling pretty buoyed by the NAFLD work, which was really encouraging. We looked to cast our net a little wider, and we had an opportunity to work on metabolomics of schizophrenia. And I've chosen a couple of artistic representations that people have drawn for schizophrenia because I think it's important. Most of us, when we hear it, this image may evoke the association that many of us first have, that we think of it as a neurologic disorder localized to the brain. But in fact, schizophrenia is a whole body disease. So clinicians and doctors have known this for a long time. And it's a whole body disease. In fact, there's really exciting research linking dysregulation of the immune system to schizophrenia. But interestingly, neurologic disorders are only fairly recently being considered with metabolomics. That's not a completely fair statement. People have considered it. But one of the things to remember is it is difficult to get draws in this case. We're discussing patients who are in a really difficult situation, and it's difficult to get draws. So that's part of the reason why metabolomics has lagged in neurologic disease overall. So to say that we were excited is an understatement because it's underserved and we have a whole body disease. People have associated schizophrenia with energy dysregulation. We thought, yes, this is going to be great. Furthermore, the maturity and the breadth of the MyCode database is so extraordinary that we can actually craft a longitudinal study of 24 pre and post diagnosis subjects. So we leapt in with both feet and we did our homework. I think we've read every metabolomic schizophrenia paper out there. There are a couple N-acetyl aspartate and 3-hydroxybutyrate. Where I want to sort of prime you a little bit, 3-hydroxybutyrate is well known to increase with schizophrenia. We were able to get greater than 50 metabolites. It was a great data set. We were going into sort of a larger part of the MyCode database where we promised to only use 100 microliters of serum. And there, the approach of using curated approaches, long acquisitions, Shigemi tubes, really worked to our advantage. So it allowed us to get into the standard of care biobanks. I think people interested in that should realize that, you know, NMR has a tremendous demand on sample volume, and we really strain biobanks. And so one of the things we need to do is we need to continue to decrease the amount of serum we draw from biobanks. Well, the first disappointment was NAA was below our detection limit. That's a shame because it's a well-known marker of many neurologic diseases, including schizophrenia. So that was a disappointment. Then one of our strongest results, ketone bodies decreased. They were supposed to increase. And in fact, they decreased pretty clearly. It wasn't ambiguous, it was significant. And this really left us with some conclusion. We had done our work, they're supposed to increase. But of course, what had happened was, if I'm being candid, I think we got so excited about the ability to study an underserved disease. We got so excited about the ability to do longitudinal metabolomics that we really didn't realize that we had actually inserted ourselves into something much more interventional, not diagnostic. And in particular, the timeline for each of these subjects was there's a pre-draw, that's our first sample. There's a time delay because at the pre-draw, they're presenting for something that's not related to schizophrenia. So it's a standard of care draw for something unrelated to schizophrenia. There's diagnosis where there's not a draw because standard of care is not a draw with schizophrenia to the best of my knowledge. There's a very short delta T between diagnosis and prescription. This delta T can even be 0. Then there's a delta T from prescription to the post-draw. And we know these, and what we came to understand was we're actually studying not the effect of the diagnosis, we're actually studying the effect of the prescription. And in fact, there is literature, actually only one, there's literature out there that shows antipsychotic drugs reduce 3-hydroxybutyrate. We're kind of excited by that finding. We're thinking of possibly getting in here and making this still something publishable. I admit I'm being a little cagey with the results. As you can see, no spectra, no data. I think we're excited that from this very, very broad population, we were able to see a clear result, but it was a real teachable moment that we had frankly gotten too excited and we hadn't thought enough about our question. Having said that, we did see something interesting, and fingers crossed we'll be able to craft a story out of that. I want to wrap up with some comments on something that's been really exciting for us, honeybee metabolomics, including bee brain metabolomics. And it follows the similar arc. We are going to do extraction because in this case, it's the only way that we can think of to test the metabolomic state of the whole bee. You can, with a syringe, extract hemolymph. That's a difficult thing to do. It has to be done by an expert. It's difficult to do reproducibly. You have to restrain the bee, etc. So we wanted to avoid that. We wanted to look at this from the point of view of whole bee NMR, and we wanted to do single bees. And a wonderful group of students and a very generous collaboratory have worked with us on this. And just want to recognize them up front here as well. And we sort of did something, so this came out of order. We actually did this before the schizophrenia study, so this is not in order. And here we leapt in feet first again. And we got lucky. We sort of saw the kind of biomarker you only dream of. It's very rare. With our colleagues, professors Beth Capaldi and Marie Pizzorno, they designed an amazing study involving deformed wing virus and laboratory-raised bees that were injected with and without deformed wing virus. And most remarkably, they were willing to dissect the brain out of the bees for us. And we were able to extract the brains and actually homogenize them, extract them, and get NMR spectra. These are 2-hour runs. It took a lot of work to get this. And we've gotten better at this since, as you'll see in a moment. But we managed to stumble across proline as a very, very strong marker in the brain. It shoots up in the brain. We took the corresponding bodies, no change in proline in the bodies, but huge change in proline in the brain. And in fact, this correlates with upregulated expression of proline-rich antimicrobial peptides in the brain. And so that's a good correlation, which made us feel more comfortable in that result. We don't know if it's a consequence of the upregulation of the peptides or if it's supplying more proline to make them. We don't know exactly how that's happening, but we're pleased to see that correspondence. Who's in the hive? Well, we are. It's a great thing to do with your students. But you want to look at workers, queens, and drones. And you can get brains for all of them, although we haven't done a queen brain, I should tell you. It's hard to get enough queens to do that. We haven't done a queen brain. Drones are big. They give us a lot of metabolites. Foragers are small. They don't give us much. But we were using the old chloroform methanol approach. And when we upgraded to the new acetonitrile approach, we were just shocked at the increased metabolite recovery. And that's great work from Jane and Sydney, who worked on that. And even though we bring along some extra sugars and lipids, it is well worth it. In terms of dissections, you want to think about how to dissect. It turns out you can keep the eyes or take them off. The main thing that we feel by comparing the recovery of metabolites with and without eyes is that you're simply recovering more brain material if you hold on to the eyes. I can't say that we saw unique metabolites by keeping the eyes. I can't say that we saw that. But what we can say definitively is we saw a major increase in recovery of brain matter and brain metabolites by keeping the eyes. So that's a recommendation that we have. Overall, the quality of spectra are outstanding. And one of the things Jane looked at was 2-to-1, 3-to-1, 4-to-1, 1-to-1, different ratios of acetonitrile. And although they're all good, there is a winner. And the winner is 2-to-1. And the reason is actually in the sugars. In the sugars, one of the problems with lower ratios of acetonitrile to water is we carry along too many sugars. But if we go to higher ratios of acetonitrile, we start to lose our metabolites. 2-to-1 is the Goldilocks ratio, where you accomplish the best suppression of sugars, but you keep the most metabolites that you can. One of the things that we want to do is get beyond dissection. It's very dependent on the dissector. It's difficult, it has to be done under a microscope. With an expert dissector, it's about 15 to 20 minutes of brain. So we wanted to look at the possibility of just cleaving the head. And so that means you just take a little scalpel and chop there. And in particular, that's going to give us the brain and several glands. And what does that look like? Well, first of all, no surprise, we see a lot for head versus body, a lot less in the head than the body. But one of the things that was fun for us was we got 10-HDA. That's a key medium chain fatty acid in royal jelly. There are several. There's also 9-oxodecanoic acid. There's several. We didn't see that one, at least at our sensitivity limit. But we're very excited to see this because the mandibular gland expresses a lot of these fatty acids that have signaling roles. We would love to get better at detecting more of those, but we're already happy that we can see 10-HDA. By the way, we did do some experiments with methanol. Long story short, they were disappointing in our hands, and we're not sure why. Sometimes methanol is very good, but they weren't good in our hands, so we're sticking with the acetonitrile. Really grateful to be working with Amy Freund at Bruker, where we're really interested in broadening the accessibility of this kind of work and maybe making different kinds of studies and questions accessible by this work. So low field NMR is so fascinating right now. We only hope to do more. So impressed with what it can do. And this is, first of all, a stability study that she did that shows that within the first few hours, there's just a little bit of variation in one signal region. And if you're willing to wait a few hours, you then get good stability. And that's a great teachable lesson from extracting whole bees and makes it possible to run a lot of bees at once. And in fact, that's exactly what's going on. If you take several bees from one hive and from another hive, first of all, the spectral quality is pretty nice, and several good fingerprint regions jump out at you. But you start to even see that there are some differences that may be hive specific. And while low field is definitely going to be better for untargeted analysis, we've been pleased to see that there are some targeted opportunities. I'm going to be a little coy because this is just emerging work, not going to sort of say what we're looking at. But we are looking at one metabolite in particular, which is showing some nice variation with hive identity. And in fact, this one is unusually low relative to the rest. And we've identified it in a stressed state, and we're wondering if that's possibly being reflected in the metabolomics. And so we have a lot more time to spend with that data. This is just sort of emerging work, and we look forward to seeing what it tells us. Broader thoughts is I think this is more broadly applicable. Here's a carpenter bee. Notice that it absolutely blows away our 0.1 millimolar DSS, and you can do carpenter bee brains. But I need to wrap up. I see the time is running short, and I want to wrap up. Overall, I'm excited about metabolomics because I don't see a lot of people working in bee NMR metabolomics. We think these are some of the first spectra to come out in that area. People have done mass spec on bee brains, including beautiful work from Dan Raftery again, worth looking at. Also, Art Edison, many people may know, he started off some really nice work doing insect NMR as well. And we're inspired to do more. But we're particularly inspired that standard of care here are very exciting, particularly because when you do find metabolites, you're finding them in broader populations. And that's been sort of one of the trope of metabolomics, that every time somebody expands their N, they start to lose significance on their markers. Instead, we're proposing start right away with very broad populations and see what survives. And even though prospective clinical metabolomics is very important, I mean, obviously we still need to continue to do it, the scope and sophistication of standard of care biobanks is really growing, and there are exciting opportunities there. And I'm just going to stir the pot a little bit. Sensitivity is important, but all of my experience, we struggle more with resolution. It's one of the things that limits us the most. So a little selling for the master's program at Bucknell, but also I just want to thank everybody we've worked with. And in particular, we're very fortunate to have the enthusiastic support of Dave Carey and Chris Till at Weiss Center for Research and the Obesity Institute there. They're a really unique group there. We have to thank Bucknell and Geisinger and NSF for funding. Lots of great folks have worked on this. One of the things when we worked with the hospital is that biostatisticians are in extraordinary demand. They're not, it's important for us to be able to enter this collaboration knowing that we can work with a biostatistician. We're extremely lucky. I get to collaborate with family, extremely lucky to have a talented biostatistician in our family who we've enjoyed working with as well. So thanks to all these folks and thanks to you, and be happy to answer any questions.
Thanks a lot, Dave. It was a great talk. Lots of cool metabolomics work. So if anyone has any questions, you can raise your hand or just go ahead and unmute yourself and ask. Yeah, it's fun.
Yes, first I wish to congratulate. This was a fantastic and beautiful talk, and a very thoughtful design of the projects. It is absolutely great. I wanted to say that perhaps the multiple access of freeze-thaw cycles may be a little rosier. The picture for that may be a little rosier than it is presented, because consistency is more important for the statistics than the absolute quantities. So if you have the same cohorts treated the same in terms of number of freeze-thaw, then the systematic changes across the set will be the same. As I recall, the changes in the measured intensities were quite systematic across the whole set of metabolites. Obviously, it may be a bigger issue if you treat things different. So you have a set which is 5 times freeze-thaw, the other is just 3 times or 2 times. And or maybe when you measure absolute quantities, although it depends what kind of standard you use for that. But again, congratulations, beautiful talk.
Oh, thank you very much. Yeah, you make a really good point about the freeze-thaw cycles. It is very systematic, and it could be built into a study for sure. And I think now that we have a little more experience with it, I think we would be even willing. We were willing beforehand, but I think I'd be even more willing afterwards to consider samples that had different numbers of accessions. I think I'd be willing to consider that now based on what we've seen. But also, you're right, since it's systematic, it's that also makes it more something you can deal with more.
Are there any other questions? All right, if not, I'd like to thank both of our speakers for this session, for 2 very interesting and exciting talks. And I think